But today what I'll do is I'll go through sort of an overview of the research we do in my group, and we focus a lot on gold nanocrystals, and I'll explain why that is. And then we're chemists, but we do experiments that touch on physics, biology, and even a little bit of ecology. And my email address is below, so you're certainly free to email me any questions that you have maybe after the presentation. So the first thing I wanted to mention is that we're nanoparticle people. And it turns out for hundreds, if not thousands of years, people have known that nanoparticulate matter has interesting properties that are different from the bulk. Of course, they didn't call it that. They called these features of matter finely divided matter. Okay, so here's an example. So here's me, in case you're wondering what I look like. Here I am looking at this cup in the Art Institute of Chicago. So this cup is called a Lycurgus cup, and I got some metal work on it, and it has glass there, which is tinted this kind of orangish rosy color here. So the Lycurgus cup was made by the Romans in the 4th century AD. It normally lives in the British Museum, but it came to the Art Institute of Chicago a few years ago, and so I took my group on a field trip to see it. And the reason for the beautiful colors here is something the Romans learned how to do, like I said, hundreds of years ago, is that if you had molten glass that you were going to pour into a mold to make some artistic object, you could take gold, the element, which you can find in the ground and it looks like gold. You can take the gold, beat it with a hammer until it's really thin and it still looks gold. And then you throw these gold foil pieces into molten glass and it'll disperse to give you this reddish color. So the pigment that was known to the Romans, they called this red gold. Okay, so even then, all right, the Romans knew finely divided metals, gold didn't look gold when it was very, very small and dispersed. Okay, we can fast forward to 1857. And if you go to London, there's a standalone research institution called the Royal Institution, and they have which has science going on in this tower. It's not associated with the university. It's its own thing. And they have a museum in the basement of artifacts of the people that used to work there. So Lawrence and William Bragg, the first guys to do X-ray diffraction, their father-son team, their X-ray diffractometer is down there. And also down there is Michael Faraday's apparatus from his lab. So he did a lot of work on electromagnetism. So his electromagnets are there. And in addition, he's the first person we know to take a metal salt and chemically reduce it to the metal in a solution to produce a colloidal dispersion of little tiny particles. And so he understood this was what was happening in his experiment. And here's his solution that's been sitting there since 1857. And you see it's got this sort of gray similar color. So in his case, he took a gold chloride salt where gold was in the 3+ oxidation state and reduced it with elemental phosphorus and carbon disulfide solvent, which is a pretty nasty kind of pair of chemicals at the time. But anyway, you can see here that this solution's been colloidally stable for a long time. So now the question is, okay, we have this concept that we take a metal and we make it very tiny and we disperse it in some medium, and now it doesn't look like the same, optical properties. And so the question is, what does that do to? So again, this has been known for approximately 100 years or more, what the deal is. And so there's a couple of ways to think about this. First of all, you can think about how far does an electron move on average in a metal before it collides with something? So that's called a mean free path. Lots of measurements in theory here, but overall, and for most metals at room temperature, we're talking 10 to 100 nanometers. So that's one critical piece of this little puzzle here. The other piece is thinking about how light interacts with matter. So you can do, if you wanted to, you could do what Gustav Mie did again a long time ago in 1908 in this case. So what did he do? He solved Maxwell's equations for a certain case, the small conducting sphere. Now, you might remember from physics, Maxwell's equations, there's 4 equations. They relate electricity and magnetism, building off the experimental work of Michael Faraday, it turns out. And you can solve these equations for cases. Just like in chemistry, we solved, the particle in a box, which is a simpler version of Schrodinger's wave equation for electrons and atoms. You can solve it for certain limiting cases. We can do that here too for Maxwell's equations. So what you need to do to solve these equations is you need to say what the metal is for your conductor. So that you need the dielectric constant of the metal, you need the size of your sphere, and you need the refractive index of the medium in which these small conducting spheres are placed. And small to Gustav Mie was smaller than the wavelengths of visible light, which are hundreds of nanometers, as you see down below. So the picture we have is that, okay, here's the electric field vector of my incoming light wave interacting with my small conducting sphere. And if I'm in resonance at the right frequency, then I will have this enormous sloshing of electrons around the surface of the particle to give you a really coherent, it's a really coherent effect that gives you this strong optical property. So this we now call the plasmon. That's not what they called it back then. So the plasmon is this coherent oscillation of these loosely bound electrons. So of course metals have loosely bound electrons. So the size scale over which this happens is a few hundred nanometers down to a few nanometers, roughly speaking. So it's one of these nano-sized effects where, again, the property of matter is different in a certain size regime. So for gold and silver, those are the metals we use the most. We don't want to be smaller than about 2 or 3 nanometers if you want these plasmons. After that, things sort of look brown. And if you're too big, like, a micron, 1,000 nanometers or larger, then your sample will just look like bulk gold or little gold flakes. Okay, so you want to be in the right size regime for this. So that's the plasmon. Now, what my lab is probably most famous for is controlling the size and shape of these little particles, so you actually move the plasmons around. So here is this, these are aqueous solutions now, about 1 nanomolar in particle concentration, so that's a pretty dilute solution. And you see here, these are transmission electron micrographs of a little bit of a heterogeneity there of what's in this sample. So these are roughly 20 nanometer spheres. And if you wanted to do Gustav Mee's calculation for 20 nanometer spheres of gold in water, you would actually calculate that they would absorb at 520 nanometers in the green, and therefore the solution looks red. Okay, but what we do is and I'll show you the synthesis in a minute, we elongate the particles, so we're making them longer and longer and longer, and their diameters are pretty tight. And so now what happens is the plasmons change on you, and that's visibly apparent in the colors here. So down here I have spectra that go with this. So here is the one sample number 1 there, that spherical sample. Here's our plasmon peak, just one of them. And then as we make a rod-shaped particle, our plasmons split into 2. So we have absorption and scattering of light along the short axis and the long axis of the particles. So the short axis band, that's called the transverse plasmon band, is pretty much still here around 520 nanometers because the particles aren't getting too much fatter. But as we increase their aspect ratio, that's length divided by width, the so-called longitudinal plasmon band, which is, you can picture the electrons sloshing along the long axis of the particles. As the particles get longer and longer, that goes to lower and lower energy or longer wavelength. Okay, so again, you can control the shape of the particle, and you control where it absorbs and also scatters light. So over here, I have this little picture. It looks like a galaxy, but what it actually is a dark field optical micrograph of our nanorods immobilized in a gel. So what we do is we take this sort of clear jello type stuff, we throw our particles in there, we put it under an optical microscope, just a regular old biology type microscope, but you can image things in quote dark field mode where you look at the scattered light instead of the transmitted light. And what you see here is all little stars in the sky are the particles scattering light like a little cone. And the misty colors are from the gel. So you can't, the field of view here is about 3 millimeters. So you can't literally say that one little spot there is one particle, but I'll show you later some data where you can actually kind of infer that. So I think it's just really cool, right? That in a bulk optical experiment, you can actually image nanoscale objects. So when I talk to students about this kind of stuff, like if they want to join my lab or something, usually the first question that comes up is, well, that's really cool, and maybe you could tell me more details about that. But the other question is, well, what is it good for? Okay, so there's applications here. So let's talk about them a little bit to set up some of the stuff that's coming up later. So, okay, so here's my particle in the middle here. So here's my little gold rod. I'm shining light. Okay, so I have these plasmons. And not only do you get these plasmons, you get a whole bunch of other stuff here. So when you're moving all these electrons around in this coherent oscillation, that sets up a local electric field near the surface of the particles. And molecules that experience that electric field will have their properties altered. So you can use this for sensing and diagnostics. So probably the most famous example here is something called surface-enhanced Raman scattering. So Raman, you might remember, is a vibrational technique where you come in with visible light, or much larger energy photons than infrared light. And these visible photons inelastically scatter off your sample to give you either picking up a little bit of energy or dumping a little bit of energy into vibrations in your molecules. And so you get vibrational spectral like you would in an IR experiment, which is a sort of a sophomore, chemistry kind of experiment. The selection rules are a little different, but the problem with Raman is that the cross-section is really small, so the peaks are really weak, and you need a ton of sample to see anything. But if you have, if this visible photon also excites a local electric field, now you're talking, and the Raman sensitivity goes up as the fourth power of the electric field experience. So here's just an example of that. So I have a little flat gold surface here. I have some molecules I've mobilized on the surface, and then I sprinkle some particles on top, not to scale, right? The particles are way bigger than this molecule. But if I just have the molecule on even a flat metal surface and I try to do this Raman measurement to look at the vibrations of this molecule, okay, for a monolayer of molecules and the spot size of my laser, there is no hope, so nothing's going on. But I sprinkle particles on of different sorts, and now these peaks are lighting up here, and so these are the ring-breathing modes of this molecule. So again, for ultrasensitive detection, people are very interested in this Sirstance-Raman effect. Up here, you can do the same kind of thing with fluorescence. So you can take a bad fluorophore, meaning it doesn't emit fluorescence very well, and you can make it brighter because it experiences these effects. But the problem here is fluorescence is usually quenched by metals due to sort of energy transfer, maybe even electron transfer into the metal. So you need a spacer layer to put your molecule far enough out that it still feels the field, but not so far out that you lose the effect. And so I'll talk about this a little bit later, but we put little glass shells on our particles here, and we're able to optimize the distance and amount of light coming out of the bad fluorophore. Okay, so the electric field is a big deal here. You also get heat when you shine enough light on these particles. The images I showed you before of our little samples, they're very dilute, right? 1 nanomolar, 1 times 10 to the minus 9 moles per liter of particles in the vial, and they have these brilliant colors. So that says they're very strong absorbers. And if you put enough light into a strong absorber and it doesn't emit light, which these don't by themselves, then you'll be generating some heat. And you can use that heat to do things. So you can get molecules to pop on or pop off the surface. I'll show you some of that. So this is things popping off, the little fluorophores popping off, so you can see them light up when they do that. And if you want to go very bio, you can use the heat to kill things. So this is called photothermal therapy. It's in clinical trials in humans right now. It's looking very promising. But the idea is that your particles, they can absorb light in parts of the spectrum that you can tune because you can change the shape of the particle. And then you can come in with a fiber optic or something. And if your particles have a high enough concentration in a certain spot, you can actually kill stuff like pathogenic cells. So here's a kind of gross example of a mouse with a tumor, and you shine light on the tumor that's got the particles, and the tumor shrinks. Now in this case, you might just say, why don't I just surgically remove the tumor? And sure, in this case you can do that. But there's plenty of other cases where it's sort of hard to get to. And so again, if you get enough particles there that absorb, especially in the near IR region of the spectrum, which you can make particles that do that and come in with a near IR laser, then the dream here is that there's no chemotherapy, right? Chemotherapy makes people really sick. The drugs go all over the place. And so if you could have very localized therapy, this would be a great thing. And again, human clinical trials are looking very promising. Okay, but to do all this wonderful stuff, we have to make these particles. Okay, so we're chemists, so we can do some synthesis here. So this is an example of a synthetic method, and we call this seed-mediated growth. So what Michael Faraday did years ago, right, he took a metal salt, the same metal salt we have here, HAUCl4, where gold's in the 3+ oxidation state, and he used a very strong reducing agent and just made a bunch of these little spheres. Okay, spheres are the lowest surface energy particle. What we need to do is control the size and shape of the particles. Okay, so to do that in one step is pretty tough, so we do it in 2 steps. In the first step, we make a little seed, as we call it. So this is everything's in water, everything's in air, everything's at room temperature for the most part. So I have my metal salt, and I have some of this stuff, this surfactant here. And I come in with a pretty strong reducing agent, sodium borohydride in water, and I dump it in there, and I let it go for about 30 minutes or so, and my bright yellow gold chloride solution turns kind of brownish. I've made little tiny gold particles, too small to have a plasma, and they're about 1 or 2 nanometers in diameter. Pretty high surface energy materials. What's on their surface is a little vague. There's all this other stuff in the pot here, and they're good for about an hour or so. Otherwise, so you got to use them pretty soon. Okay, so you make them, and then in the meantime, you make a fresh pot with more metal salt and some other stuff here. And then what we're going to do is take this solution, add a weak reducing agent. Hydroquinone is what I'm showing here, but we also use vitamin C, ascorbic acid. So you mix all this stuff up, and I'll explain what these things are in a minute here. we add our weak reducing agent, and then we add these little seed particles. And the idea here is we're using a weak reducing agent because we don't want to have all our metal make more metal seeds. We want to have the metal deposit on these seeds and then grow. And that's because the redox potential of the free metal versus an adsorbed metal is different. So the idea is that the seeds will help nucleate the growth on them, and then they will grow. Okay. So if you just do this, HAUCl4, weak reducing agent seeds, you'll make spheres, okay, that are bigger than these guys here. If you want to control the shape of the particle, that's where these other things come into play. And now it's pretty important what you have down here. This was sort of optional, the CTAB stuff. Now you need a lot of the CTAB stuff in water, and you have to have an additive metal ion impurity at a low concentration here. Here it's silver nitrate is our additive metal ion impurity, and operationally a little bit of silver nitrate gives you a short rod, and more silver nitrate gives you a longer rod. Okay, so very interesting chemistry questions about how does that work and all that kind of stuff, which I won't get into too much. But suffice to say, this is how we control the growth of the particles by changing our reagent concentrations. When you're done, you do have a little bit of silver in these rods, and you can quantify that. Okay, so we make this stuff, and they're gorgeous. Okay, so we do a ton of transmission electron microscopy in the group. So these are the little rods with a little bit of silver. You add more silver, and really monodisperse compared to what you might expect would be happening in solution. Okay, so we love it. All right. So that's what we're probably most famous for. And even though we've been doing this for a long time, we keep evolving and getting better. So the particles, as I just described, using, in this case, ascorbic acid as our weak reducing agent, the particles come out to be somewhere like this, maybe 15 nanometer-ish diameters, and then you can tune the length 30 through 80 nanometers. That's about the size of a virus, in case you're wondering how big that is compared to some natural object. But we also wanted to be able to control the absolute dimensions of these particles. So my student Wei Wei Chang at the time, published a couple of years ago now, came up with this modified method to have the plasmons still have the same rainbow, okay, but have the absolute particle sizes be smaller. So that's one of these things that sounds easy to do. Well, the concept is easy, but then how do you do it is actually pretty tough. So she came up with two things. One thing is you just add way more seeds here. So now for the same amount of metal precursors, obviously the final particles are going to be absolutely smaller. But to control the growth well, it turned out we needed to add some HCl at the front end here. Interesting chemical story again. You might think, oh, what am I doing here with HCl? I'm changing the pH. Well, actually, you're doing a lot more things than that. That's one of the frustrations of people that work with these colloidal materials. It sounds simple to dump everything together and stir, but it turns out there's a lot of timing issues, and changing one reagent at a time actually changes a lot of things. So just as an example here, ascorbic acid, our weak reducing agent here, is an acid. Its redox potential, which is important for this reaction, of course, depends on pH. So you're changing that. The other thing you're doing is you're adding a bunch of chloride, okay? And up here at the front end, we have a lot of bromide counterion, and we have a lot of chloride counterion. And gold, chloride, and bromide, they can exchange, and the redox potential of the gold, bromide, or chloride is different, and it's different for different mixtures of chlorides and bromides. So you're actually changing lots of things when you change just what looks like one reagent at a time. But anyway, just an example to show you that we can tune things a lot here. Now, when we're done making all these particles, what do we have on their surface? They're colloidally stable in water. And I mentioned we have a ton of this surfactant here. So the question is, what's on the surface? Okay, so we have lots of lines of evidence that we actually have this kind of structure. So here's my metal core, a little bit of silver in there, as you see. We think the first thing that sticks to the surface is the bromide counterion of this surfactant, which we hadn't thought actually initially would be important at all. But if you change it out for chloride or iodide or other anions, you actually don't get any rods. So the bromide's pretty important. And then we have electrostatic ion pairing interactions with the quaternary-ammonium head group here, okay? And in water, this is a very hydrophobic tail, so that's not stable, but we have so much of the surfactant around, we end up making a bilayer. So again, lots of lines of evidence to say that we actually have this kind of structure. So it's pretty interesting, right? It's about a 3 nanometer hydrophobic region next to a hunk of metal, and highly positively charged groups facing the solvent here. So once you realize this stuff is there, then you can start thinking about doing chemistry on it, right? You can try to stuff small molecules in here, try to replace this whole thing, etc. Okay, so that's sort of a little intro to the particles. And so I have 4 little short stories here about things you can do with them. So the first one is sort of more physical, PChem kind of stuff. And here this goes back to our plasmons. So here's, let's say, the surface of my particle. Here's distance from the particle. And if I shine light on my particle where it absorbs and I want to measure this electric field, how far out does it go? Well, it's hard to measure directly, you might say. So you can certainly do a lot of calculations. Everybody would say, well, after about 150 nanometers away, you know, you're not going to feel anything if you were a molecule trying to experience this field. In fact, people would say it's probably closer to 50 nanometers. Okay. So I mentioned earlier that if you have a bad fluorophore, meaning it doesn't emit light very well when you shine light on it. So a lot of fluorescent dyes, of course, are very good fluorophores. You put in a little bit of light, they glow green, orange, blue, etc. A lot of dyes in the near IR, again, for bioimaging, that's a nice place to be because you get out of all the interference from tissue. A lot of near-infrared fluorophores have weak fluorescence quantum yields, but you can boost it if you put them near one of these plasmonic particles. So the challenge here is that you can't be, like I mentioned before, you can't be too close and you can't be too far. So if you're too close, you'll just quench and you don't get any light out. And you do have a surface enhanced fluorescence effect, again, due to these, there's some equations down there in the bottom, which I'm not going to show you, about how the cross section for absorbance depends on local electric field to the second power in this case. Anyway, if you look at the literature, what you find is that people experimentally find that somewhere between 10 and 20 nanometers out, your bad fluorophore has more light coming out than you expect. So you're balancing these two effects, enhancement and quenching as a function of distance. And then the question is, how much more light do you get out of your bad fluorophore? This is where the literature has been confusing. Factor of 2, factor of 10, factor of 100, factor of 1,000,000, okay, depending on your measurement. So we decided to dig in here a little bit. The question is, how do you get your fluorophore a certain distance from the particles? So there's lots of ways to do that. One thing we do a lot in the group is we do layer by layer polyelectrolyte wrapping. So here's my original little broads. And you can come in with commercially available anionic or cationic polymers at the right pH, of course. And you can incubate them and purify them, and then you build up layers here. And we've done lots of measurements like this. The thing you measure most easily here is something called a zeta potential. That's the effective surface charge. So a big positive number means it's cationic, and a big negative number means it's anionic, and 0 means you don't have a net charge. So we can, monitor all these processes by measuring the zeta potential, which is a bulk measurement. So what we did in this experiment with the fluorophores, though, is use a silica. The layer by layer polyelectrolyte wrapping, each layer is roughly 1.5 nanometers when you add it on there. And we needed to get 10 to 20 nanometers out, and that's a little bit of a job. So here we're going to grow little glass shells. So again, we have chemistry here to do that. I won't go through that too much. But then here's our dye. It's a pretty big molecule. It's got a little carbon-carbon triple bond right there. And we're going to do something called a click reaction to react this carbon-carbon triple bond with an azide. So we had to get the azide on the glass. So we had to do some chemistry here to get this silane on the azide. And then we mix everything together and do our little click reaction. And this dye absorbs and emits in the near infrared, so past the red region of the spectrum. And of course, we can make particles that will overlap this really well. So in the big experiment here, we're going to make the particles that have plasmons that are on the blue side, on top of, and on the red side of where the dye does its thing. The hypothesis here is that you want the plasmons to be on top of both of these things. And the plasmon's pretty broad, so it will hit both of them. And then the variable we weren't so sure about is how thick should the silica shell be, okay? But somewhere between 10 and 20 nanometers is the idea. Okay, so it took a while to figure out, but we did figure out ways to control the silica shell thickness. So 6 nanometers, 11 nanometers, 12 nanometers, 15 nanometers, et cetera here. And here's our particles that are absorbing on the blue side, on top of, and on the red side. So that's a lot of samples here. And we had to quantify all the density of these molecules on the surface. Okay, there's a lot of work there too. But in the end, what we find is the following. So I'll just draw your attention to the maximum bang for our buck here. So what we learned is that 17 nanometers out seems to be the magical distance. And also the plasmons, I didn't explain it explicitly here, but the plasmons being on top of both the absorption and emission of the fluorophore is where you want to be. And then how much more light do you get out? It's about a factor of 10. Okay, so that was a very thorough study. And for this kind of work also, you want to make sure that there's nothing weird going on, so you do some time-resolved measurements as well. And it's more complicated than I thought in terms of the photophysics, but I'll kind of skip that for right now. But if you think about it, This actually doesn't make a lot of sense because when you shine light in the near IR where these particles are absorbing at their longitudinal plasmon band, if you do a calculation of this local electric field, it is not uniform over the surface of the particles. The ends are where the field is the highest. So what this says, and we can't control, okay, where the molecules go. So what that says is that the actual enhancement is probably way more because only molecules that happen to be on the ends are going to experience this enhanced field and give you more light out. And all those molecules in the middle, which is most of them, right, aren't going to do anything. So that means we actually have to get good at doing something like this, which is to say, making particles and functionalizing their surfaces in an anisotropic manner, meaning not just all uniformly all over the particles, but just on the ends or the sides. So here we have some silica. So we had silica glass all the way around before, and now we've gotten pretty decent at making it just around the middles of the rods or just at the ends. And this is EELS, electron energy loss spectroscopy. It's a technique you can do in a transmission electron microscope to do elemental analysis. And so here we're actually looking at carbon, silicon, and oxygen. So the silica looks purple because we've got, it's SiO2. So we got both silicon and oxygen in the same place, but the carbon is from our surfactant that is in the pores so we can actually visualize that. So I wanted to say a little bit of thing about the heat. So you might remember from general chemistry, that, Le Chatelier's principle, right? If you stress a system, it'll, go back the other way, right? To re-equilibrate. And in the world of drug delivery, it turns out people do use these plasmonic heating effects. And the idea is you have your particle, it's got maybe some drug you want to deliver, at the right time. And so you put your thing in the system and you come in with light and then you shine light, generating heat, and then the molecule will pop off. Okay, so that's general kind of thing people do. But if you think about it, that's only going to work for a certain situation. So here I've got, let's not worry about what these mean here right now. Let's say I have A plus B makes a complex here. And if my reaction is exothermic to make this complex, and I increase the temperature, then you would, according to Le Chatelier's principle, right, you should decomplex those two things. So in the case of an exothermic association, yes, shining light should generate, which would generate heat, would make things pop off the surface if one of these things was my gold particle. But if the fundamental thermodynamics of the system is endothermic, that is to say, A plus B plus heat makes the complex, and you shine light to generate heat, now you're going to make more stuff sticking to the surface, which is actually the opposite of what you want if you want to do delivery. Okay, so here's the question is, let's see if that's true. So what we're going to do is take our particles, wrap them up with these polyelectrolytes, okay, and then what BSA and lyse are, these are proteins. So the hardest part of this project was actually to figure out what proteins and what polyelectrolytes fundamentally have endothermic or exothermic interactions. So we've dug in the literature to find that. But here's the concept. I wrap up my gold rods with the polyelectrolyte. I come in, I have a solution of them with protein. I shine light. And then in the endothermic case, I actually adsorb more protein to the surface. And when I centrifuge off the particles, now there is less protein in the solution than when I started. It's a very old school experiment. There's nothing super fancy here. And then in contrast, if I have the exothermic situation, I have my particles that absorb at a certain wavelength of light. I wrap them up with the polymer. Here's the protein that is an exothermic association with that polymer. I shine light. Now stuff pops off the surface. And now when I separate out the particles, I should have more protein in the supernatant than what I had before. Okay, so that's very old school. So yeah, we can do that. So here's our particles. So we have a certain laser. We make the particles so they absorb at that laser wavelength. Again, very dilute solution. And up on the top here, you can put a thermocouple of the solution and measure that. Sure, in just a couple of minutes, you're changing the temperature by 10 degrees. That's plenty to do biotype stuff. Down here, we got to confirm we're still endo and exothermic. So BSA is our protein and PAH is one of these polyelectrolytes. That's the endothermic case. Lysozyme is the protein over here, and PBS is another electrolyte, polyelectrolyte. That's the exothermic one. So this is isothermal titration calorimetry data. You can do in this under these conditions here, and these little downward spikes and upward spikes means endo and exo respectively. And okay, for the polymer plus the protein alone, the endothermic case is 400 kilojoules per mole positive. On the particle, it's still positive, but you see we lost a lot of numbers here, so it's going to be a much more mild effect. And on the exothermic side, all by itself, these guys are minus 64 kilojoules per mole, but when you put them on the particle, okay, you're still negative. That delta H being negative is exothermic. but it's not as big a magnitude. Okay, so we're kind of asking a lot here of these little particles. But nonetheless, when we do the experiment, it actually works. So here's the endothermic case. So we do, in fact, pop on the particle. So now there's less in the supernatant afterwards. And then in the exothermic case, we pop off protein. And so yes, afterwards, there's more in the supernatant. 2 independent assays to quantify all that. So yay. But then we decided to push it. So here's what we're going to do. We're going to do the same experiment. Instead of just a nice buffer solution, we're going to do this with a mix of proteins. Okay, fetal bovine serum, which has like 5,000 proteins, from, at high concentration here. So we have to label our particular protein so we can measure it, okay, in the supernatant. So we have a fluorescence tag on the 2 proteins we had before. And what we find is even in the context that's much more biological of all this soup of proteins, you can still predict what's going to happen. So you do pop on a little bit and you pop off even more. Okay, so fundamentally the notion here is that depending on the delta H, the enthalpy of interactions here, you can kind of control and predict what is going to pop off the surface of these particles with whatever surface coating and what will pop on. So in other words, you can control local concentrations of proteins, okay, over some time frame with this kind of idea. Well, so that's the sort of more physicsy end of things, okay, looking at the heat and the electric fields and so forth. So I want to talk a little bit about the surface chemistry. Okay. So you might have noticed this cartoon I had, right? I had this cartoon that, showed these molecules on the surface, okay? But you notice I've also kind of implied that there might be a different density of them on the sides versus the ends, which is one reason that you might actually be able to put stuff just on the sides or the ends. Okay, the question is this true? Is there really less stuff on the sides than the ends? It is really hard to make these measurements. So what you have to do is you have to do individual particle measurements, and you have to do elemental mapping and quantify stuff on the sub-nanometer scale. Okay, so that's pretty tough. So what are we going to do? We need help. Okay, so Pinchane Huang, who's part of our I-MRSEC as well, and Blanca, her student, part of our I-MRSEC as well, are going to help us out here. So they do a lot of STEM EELS. That's skinning, transmission electron microscopy, and this elemental analysis technique I mentioned before. So here's what we're going to do. We're going to put these particles that have this stuff on it, this exact stuff, this is stuff we care about, on a single layer graphene grid. We're going to use the electron beam as kind of like our probe, and we're going to quantify how many carbons are in the spot size of our beam on the graphene, and we know that, of course, how many carbons there are in a single layer of graphene, so that's our calibration. And then we move over to the particle, and we go all the way around and quantify number of carbons relative to that of the graphene. And then we do that, 100 times, okay? And then we get a number for how many carbons are there on the sides versus the ends. And the question is, do we actually have a measurable difference here? Lots of indirect experiments suggest that that would be the case, but no direct measurement. Okay, first problem. We have to be in water for this experiment because our particles with that particular little bilayer on them are not stable in anything else. Here's the problem. Well, when you normally make a transmission electron microscopy grid, okay, you normally put your solution on and just let it dry, right? No problem. And people know how to do this. There's all kinds of little tricks you can do, and there's drying effects and whatnot. But the problem is water has a very high surface tension, and on a single layer graphene grid will crumple the grid, and therefore you can't do any measurements. So, okay, that's kind of an annoying problem to overcome. So here's what we did. We came up with this little ultrasonic spray system. So I had a student in my group named Josh Hinman, and he has a twin brother named Jordan Hinman. And Josh was in my lab, and Jordan was in Ken Sussex's lab. Ken Sussek does a lot of ultrasonic spray pyrolysis. The two of them work together to build this little system where we have an ultrasonic nebulizer. This is like what's in a water humidifier. And it produces little tiny droplets, okay, that you can push with a carrier gas through this system and heat up things if you want to up here in the drift tube. And the idea is to gently deposit on your single layer graphene grids, which, by the way, are not that easy to make anyway, but Blanca and Pinchane know how to do that. Okay, so the hope is that the grid will not crumple, so you can use it to calibrate your carbon signal when you do this elemental mapping. And indeed, we managed to get it to work here. So sample A, there's all these little holes here. If you see black, that means there was a single layer graphene, and then gray is, yes, you have single layer graphene. So if you just cast a drop of water you ruin all this graphene, so forget it. And here, if you do this little spritzer deal, okay, a lot of them survive. That's these little gray ones here. And then you can zoom in and zoom in and zoom in, and you see the metal, and then you see some other stuff here. Okay, that's the stuff we care about. And as a point of fact, we also noticed when you just do a drop cast, of course, you get a random sort of array of particles here. These are silica-coated gold nanorods. And if you zoomed out, you'd see some clumping. You know, that's just sort of standard. But with these little droplets that you do in the spray deposit method, you actually get very uniform. Well, from a distance, it looks pretty uniform. If you zoom in, there's little clusters of them. And it turns out the clusters depend on the particle concentration, and you can make calibration curves and all that stuff, which is kind of fun. So in other words, if you have no idea what your particle concentration is, you might have an estimate of it if you look at how many particles in a cluster you have. Turns out there's an equation from the ultrasonic nebulizer literature, of which I was previously not aware, that predicts how big your droplets ought to be, and we did some experiments to confirm that that is the case. You need to know the surface tension of your liquid, which is water for us, the density of the liquid, which is water for us, and the frequency of your ultrasonic nebulizer. Okay, so now we're ready to do the real thing. We can go ahead and look at carbon. You can look at nitrogen. There are some nitrogens in that molecule, but there's a lot fewer than carbon, and you can't really calibrate the nitrogen very well. But when we go ahead and do this and calibrate on the graphene grid, okay, here's some raw data, and yeah, indeed, it looks like there's an anisotropic distribution of carbon. That is to say, there is more carbon on the sides than on the ends. And again, individual particle measurements just to show you that this is a lot of work and there's a lot of scatter in the data. So we go around and around and around on a bunch of particles. And overall, what we find, it's probably easy to look down here, we see about a 30% dip at the ends compared to the side, which is actually a lot bigger than I thought we would see. But again, there's a lot of scatter in this data. Now, at this point, we had an interesting question. What kind of control experiment can you possibly do here? Because Pinshane told me, well, in my field of electron microscopy, people will say, you're coming in with an electron beam on a little piece of metal. How do you know you're not just ablating molecules at the ends, at the tips of the rods, and they're destroying stuff so it looks like there's less on the ends, but in fact, your measurement has messed that up. How can you control for this? I'm like, oh boy. Well, it turns out we had done other experiments in the group on these mini rods, okay, with this particular ligand. So this one over here, the original guy, this is the CTAB, that makes a bilayer on the surface. This very similar looking molecule has a sulfur at one end, and that binds to gold to make just a monolayer. And we did a bunch of other experiments on these mini rods, which made it, again, indirectly concluded that, it looked like mini rods with this molecule don't look like they have any. It looks like it's uniform all the way around from what we could tell. So those particles we put in did all the same thing. And indeed, those particles don't show any difference at all. We still don't actually have a good prediction as to why this would be, but the measurement is now very clear that you can't actually, first of all, you can measure this at all, which is kind of a triumph. And now the question is, can we go forward and try to understand what the difference is here? But anyway, we were very happy to see that we could actually come up with a control experiment that also had little metal rods. Okay, I only have about 20 minutes left, I guess, and I have a whole bunch of other stuff I want to share, so I'm going to have to accelerate a little bit. So we've also got interest in sort of the more biological side of things, and so one thing that we're interested in is proteins that are involved in neurodegenerative diseases. Okay, so Alzheimer's is probably the most famous disease there, and that's the A-beta peptide, or the tau protein you might have heard of in the news. Alpha-synuclein is the protein we care about here, and that's involved in Parkinson's disease. So if you look in the brains of Parkinson's disease patients, they have all these fibrils, okay, which are this protein, alpha-synuclein. And it turns out alpha-synuclein is pretty abundant in the brain. People do not know its native function, which is interesting after all these years. But what they do know is that there's this correlation of fibrils in this disease, Parkinson's disease. It also seems like, just like the A-beta peptide for Alzheimer's, that it's not really the fibrils that are neurotoxic. It's probably more small aggregates of proteins that are actually toxic. And so then to the cells, I mean. So the question is, can we control what alpha-synuclein does when it starts to aggregate and then grow into fibrils? It's also a biophysically interesting protein all by itself. It doesn't have much of a structure in solution, but if you put it in the presence of 25 nanometer phospholipid vesicles, it goes alpha helical, one of the classical protein secondary structures. You can tell that by this technique called circular dichroism here. And if you put it in the presence of small primary amines, it goes beta sheet, again, detected easily by this optical technique. And the fibrils are beta sheet mostly, so the idea that this would be sort of, not good, whereas the alpha helical structure might be related to its actual function, which still people don't know. Well, you can see where this is going. Whenever we hear that there's some protein that changes its conformation when it binds to some nanoscale object like those little vesicles, okay, we get very interested. So here we just need spheres, so we're going to make some gold spheres, control their surface chemistry, incubate them with alpha-synuclein, and we're going to build up layers of this protein on the surface. And up here, what we want to figure out is the overall binding constant for the so-called soft or outer layer and the harder, more tightly bound inner layer. Lots of ways to do that, which I won't get into too much. And then the question here is for this bound stuff, the protein that's bound to these little vesicle-sized particles, are they alpha helical or beta sheet, the protein that is? Here we have a problem. The problem is that particles absorb a lot of light, and they actually interfere with that optical measurement to conclude whether you have overall alpha helix or beta sheet. So we had to come up with another technique, which basically involves chewing the protein with an enzyme and doing some analysis of the pieces to see what part of the protein is facing up. Okay, so that's the measurement here. So we can get binding constants, and you can do that by dynamic light scattering, and also the plasmon band will shift as you add protein. That's another nice feature of the plasmon band. You can use it to do some sensing itself. So our reddish spheres are slowly shifting a little bit more to the red. And so we can get numbers here for how many proteins are bound and all that stuff. So that's kind of good. There's a pretty big difference here. These are the biologically relevant sizes, and you have a few hundred proteins overall bound, and most of them are bound really tight. And for a much bigger particle, only about a tenth of them are bound pretty tight. Then we do this whole chewing thing, okay, which I won't go into. But the idea is what we learned, that there's a preferential orientation of this protein on the surface. The N-terminus, which is most of the positive charges, is bound, it's going head down, so to speak. And the particles we started with initially had a net negative charge. And here's all the positive charges in the protein. They're mostly at this end. So then you think, oh, well, that was easy. I could have predicted all this just on simple electrostatics. Well, if that's true, you should be able to do the same thing with a cationic particle and predict what would happen. And so here, this cationic particle has got all these amines, as you see. And remember, amines make this protein go beta sheet. So yeah, that's what happens. You try to do these experiments to get binding constants and all that stuff, and it's a total disaster because the protein starts aggregating and forming all these beta sheet structures, and it starts precipitating out, and it's a big mess, okay? So that says electrostatics ain't so easy here. So what we then were able to do is to do different kinds of experiments where we have not primary amines, but quaternary ammoniums. So these are methyl groups here. So it's not nitrogens with hydrogens, but nitrogens with CH3 groups, still positive charges. But that positively charged surface gives you better behaved particles. You can actually do all the same binding experiments and so forth. And we can do NMR measurements here on the protein, which is a much bigger deal and much more clear about what's going on. And overall, I'll just say that we can flip which way the protein is oriented. So on the negatively charged surface, the protein does what it did before. In terms of the nitrogen head is down, the amine head is down. And if you change the surface chemistry of the protein, now you flipped the orientation. Okay, so something like this. So this becomes very interesting because, again, if you want to control what this protein is going to do, now you have a way to at least control which way it's pointing on a little nanoscale surface. And so now we're in the process of doing a lot more experiments, on this protein. Okay, so I'm going to just tell you two more little things here. I mentioned, way long ago that the particles scatter light and you can use them to do imaging. So here's an experiment where we have, we're going to look at the cellular level now with these particles. So we have a little dish, we have a soft substrate on a dish, we put cells on the soft substrate and let them crawl around. And then we put some particles all over them. So particles are everywhere here. And here's just a little movie of how we're going to look at the cells moving around. So the cells aren't labeled with anything. They're not dyed, so it's kind of hard to see them at first. But what's going to happen in this movie is they're going to get brighter and brighter and brighter as they, quote, suck up all these particles, okay? And the particles scatter light and are visible in this measurement. And as they are concentrated, it's easier to see them. And you'll see dark trails left behind where the particles were. because they were all over the place and the cells kind of like sucked them up. So we call this the vacuum. Now, one problem with dark field optical microscopy is that anything that scatters light shows up. So there's going to be little bits of salt or something floating around in there. So these cells, they're human dermal fibroblasts. They're nothing fancy. They're just sort of generic cells. That looks like there's a big hole in the cells. That's the nucleus, which is pretty large. And you see the cells, you know, if we wanted to believe these two-dimensional images of three-dimensional objects, we would say that, you know, oh, the particles don't look like they're going in the nucleus. Okay. All right, well, this is pretty fun. Okay, you can do this a lot. You can change the particle size, you can change the particle charge, you can change, okay, you can change the cell type, you can do lots of experiments here and measure these individual cell trajectories. You can scrape the cells off after this and measure how much gold they have. That's actually pretty easy to quantify, and it's a lot. They seem to suck up the stuff a lot. I mean, if I was a cell, I wouldn't want to eat all this heavy metal, but hey, that's what they're doing. So what's interesting here is that you can look at the individual cell trajectories. And so let me just focus on the middle data here. So this is the mean square displacement over time. So this is how fast the cells are moving. So here I have, this is an average of a bunch of cells. So here I have these PC3 cancer cells. Black is no particles. If I feed them all different sorts of particles, anything I feed them slows them down, which makes sense, seeing as how they're taking up all this extra mass. But here are these human dermal fibroblasts, these supposedly normal cells, and black is no particles. And some particles make them slow down, and some make them speed up, even though they're taking all this extra stuff up. So then, okay, so we're changing cell behavior. So then we got to do a lot of biology stuff here and figure out what's going on. So we do all this gene expression stuff. And the only thing to get at it here, looking at 14,000 genes, is that, first of all, going from one cell type to another, hard to predict what's going to happen. And also that lots of stuff is changing. So this sounds bad, right? It sounds like things are, you know, particles are going to kill us all. But it turns out that cells can adapt to this kind of stress. So here's a quick experiment where we incubated these human dermal fibroblasts for a really long time with a really low dose of a bunch of particles. Okay, so that's this chronic situation. So how long is long? Usually people do cell experiments for 28 hours or 24 hours or 48 hours. Here we're doing it for 5 months. It's hard to keep the cells alive that long. Or we can go ahead and incubate with just a low dose of these particles for a day and then let the cells go for 5 months. And we can quantify uptake and stuff. And what we find is that the cells they survive. They're not super happy. They got a little bit of stress. But what's interesting is the 5-month exposure of this low dose the whole time. There's numbers underneath here, which I'm not showing. The cells actually adapt to these exposures. So that's good news, right? The cells, they get stressed. They're stressed out if they only see the particles for a little while and they don't see them at all. They still remember the stress 5 months later. But if they have that the whole time, they will actually adapt. And the last thing I want to mention is going all the way out to the ecological level. That is to say, if we put our particles in some natural system, like the estuary, that's where the river meets the sea, where do they go? Like what organisms do they end up in? Do we, like you might remember, mercury is supposed to accumulate in fishes or something like this. Okay, what do these particles do? So there's facilities around the country that are federal facilities to do experiments like this. So we went to the southeastern United States and did an exposure of our particles to, they pump in water from the harbor into these tanks in this little facility, and the tanks have all kinds of critters, and there's natural waters, and we only got 1 dose, okay, here, which is again a low dose, and 3 replicate tanks. And we really weren't sure what we were going to find. So this is a lot of work here, but what we ended up finding is that in the living food web, by far the biggest sink for these particles is the microbial biofilms, right? Roughly 2/3 of the stuff ends up in the slime, okay, the biofilms. People thought that maybe, oh, it goes in the soil, it'll go in the grasses, and nope, not so much. Okay, so this is just one type of our little particles, our original little CTAB-coated rods. And then we did everything all over again and did one of those rounds of polyelectrolyte coating. So this is now we had a positively charged particle, now we have a negatively charged particle, same size, same dose, and we do everything all over again. And this is a 2-week experiment here, and then we spend 8 months analyzing the data. And everything is different. So even though you'd think after 2 weeks, who knows what's going on with these particles, right? But even though the only thing we changed at the front end was the surface chemistry, the distribution in the ecosystem is actually pretty different, right? The plants are coming online. The biofilms are sucking up even more, okay? The clams are going down, et cetera. Okay, I'm not an ecologist, so people who are eco would get very excited about this kind of data here. And most recently, we've been looking at these microbial biofilms, right? This is where a lot of bacteria are. And it turns out when we do exposures in a very messy system here within a bunch of environmental engineers, the microbial community population actually, again, responds. So here we're looking at gene sequences for thousands of different kinds of bacteria in this messy system. And you put in a little bit of our particles and the whole community, they're not huge changes, but the idea is that some species are less abundant and more species are more abundant after an exposure of, again, these most innocuous kinds of particles. So this is a very interesting place to be right now, right? Thinking about sort of long-term effects of these little particles in biological systems. Okay, so let me conclude real quick here because I've got a 10 o'clock meeting. What did we learn? All right, on the physicsy side, okay, these plasmons are pretty cool. And again, molecules that experience the plasmonic field, you can get bad fluorophores to light up. You can do some Raman measurements of molecules. You can pop things on and pop things off the surface using the heat, okay? And Le Chatelier's principle works. Maybe the only time in the last 100 years anybody's done an experiment about Le Chatelier's principle. On the chemistry side of things, right, adsorbed molecular species like our alpha-synuclein protein, right, they may not adopt the same shape as they would in the wild, so to speak. And we have measurements now where we can look at how molecules are displayed on these surfaces. And again, we're doing a lot more quantitative molecular imaging on these particles. That's the STEM EELS stuff that we're doing with Pinchain. On the bio side, doesn't sound good, right? Cellular behavior and genes are changing in live cells upon exposure. But again, over time, cells will adapt. So that's kind of a helpful thing, I think, to think. And then finally, on the eco side, the surface chemistry, even though we don't quite understand how, apparently does influence the environmental fate of these particles. And again, microbial communities where you got many, many species of bugs, so to speak, they shift in response to this exposure.